Welcome everyone. I'm excited to kick off study session 4 today, focusing on fatty acid oxidation. This is a crucial topic in understanding energy metabolism. On this slide, you'll see the title of our session. We'll briefly explore how fatty acids are broken down for energy, the key pathways involved, and some important clinical implications. I'm looking forward to a great discussion, so let's get started. Welcome to this section on learning outcomes. Here, we'll focus on understanding the key processes involved in fatty acid metabolism. We'll explore fatty acid activation, which is the first critical step, followed by the oxidation of fatty acids that leads to energy production. It's important to grasp the net ATP yield from palmitate oxidation, as it highlights the energy efficiency of fat as a fuel source. We'll also look at how these concepts tie into the overall metabolic pathways. Additionally, I will introduce some tutor-marked assignments, which can be done individually or in groups, to reinforce your understanding. Finally, we'll wrap up with a summary and self-assessment questions to test your knowledge and ensure you've grasped the main points. This slide sets the foundation for understanding how fats are broken down and utilized for energy, which is a central theme in metabolic studies. Fatty acids serve as a more efficient source of energy than carbohydrates. This is because they are reduced and anhydrous, meaning they contain more energy per unit weight. In fact, the energy yield from just 1 gram of fatty acids is about 9 kilocalories, compared to only 4 kilocalories from the same amount of carbohydrates. Since the hydrocarbon portions of fatty acids are hydrophobic, or water-repelling, these molecules can be stored in a mostly anhydrous, or water-free, environment. In contrast, carbohydrates tend to be more highly hydrated, which means they hold onto more water. If our bodies relied solely on carbohydrates to store energy, we would need to carry around a massive amount of water along with the glycogen. For example, to have the energy equivalent of just 5 kilograms of fat, a person would need to store about 31 kilograms of hydrated glycogen. Hibernating animals, like bears, provide a great example of how fat reserves are utilized efficiently. During their long hibernation periods, sometimes lasting around 7 months, they rely heavily on their fat stores for energy, rather than on carbohydrates. This highlights just how effective fat is as an energy reserve, especially in situations where conserving water and maximizing energy density are crucial. Let's take a closer look at how fatty acids are used for energy production. The key thing to remember is that this process varies a lot depending on the tissue and the metabolic state of the body. For example, whether we're fed or fasting, resting or exercising makes a big difference. In the heart and skeletal muscle, fatty acids are a major energy source. They really rely on them to keep functioning. But the brain is different. It doesn't use fatty acids as efficiently because it has limited ability to transport them across the blood-brain barrier. Red blood cells are also unique. They don't have mitochondria, so they can't oxidize fatty acids at all. Instead, during prolonged fasting, the liver steps in. It takes acetyl-CoA, which is produced from fatty acid oxidation and amino acid breakdown, and converts it into ketone bodies. These ketones then become an important fuel source, especially when glucose is scarce. So, understanding these differences helps us appreciate how the body adapts its energy use depending on the context, which ties into the bigger picture of metabolic flexibility we're exploring in this presentation. After studying this session, I expect you to be able to describe how fatty acids are activated and how fatty acetyl-CoAs are transported into the mitochondria. We'll also go over the steps involved in beta-oxidation. Additionally, I want you to be able to calculate the net ATP yield from palmitate oxidation. Finally, we'll discuss in detail the concept of fatty acid oxidation itself. These learning outcomes are essential for understanding how fats are used as an energy source in the body. Let's make sure to connect these points to the bigger picture of metabolic pathways and energy production. When the body detects a low blood glucose level, hormones like epinephrine or glucagon are secreted to respond to this energy deficit. These hormones activate a cascade inside the cell, beginning with the phosphorylation of hormone-sensitive lipase. This enzyme then breaks down triglycerides stored in fat tissue into glycerol and free fatty acids. The free fatty acids are released into the bloodstream, where they bind to serum albumin, a protein that helps transport them efficiently to tissues that need energy. Once they reach their target tissue, the fatty acids are released from albumin and move into the cell's cytosol. In the cytosol, fatty acids that are destined for energy production are initially activated. This is a crucial step, before they are transported into the mitochondria, the cell's powerhouse. Inside the mitochondria, these fatty acids are broken down into acetyl-CoA, and during this process, NADH and FADH2 are generated. These molecules then enter the mitochondria's energy-producing pathways, specifically the TCA cycle and oxidative phosphorylation, resulting in the production of ATP, the cell's main energy currency. This entire process, starting from hormone signaling to fatty acid oxidation, highlights how our body efficiently mobilizes and uses stored fat to meet energy demands, especially during fasting or exercise. Let's take a closer look at how fatty acids are activated for oxidation. First, ATP is hydrolyzed to AMP and pyrophosphate. This step is crucial because it provides the energy needed to activate the fatty acid. The pyrophosphate is then further hydrolyzed into two inorganic phosphates, or pi, which drives the reaction forward. Importantly, this process consumes two high-energy phosphate bonds, highlighting how energy-intensive fatty acid activation is. Now, the enzymes responsible for fatty acid oxidation are located in the mitochondrial matrix, which is the powerhouse of the cell where these reactions happen. Since fatty acetyl-CoAs are generated in the cytosol but need to be processed inside the mitochondria, they must be transported across the mitochondrial membranes. The inner mitochondrial membrane, as you might know, is impermeable to CoA and its derivatives, so they can't just diffuse through. Instead, there's a specialized mechanism that allows fatty acetyl-CoAs to enter the mitochondria efficiently. This transport system is essential for maintaining the flow of fatty acids into the mitochondria, enabling their subsequent breakdown and energy production. So, in essence, the activation of fatty acids is a carefully orchestrated process that requires energy input and specialized transport, ensuring that fatty acids are ready for oxidation in the mitochondria. Now, let's look at how fatty acyl groups actually enter the mitochondria, which is essential for their breakdown and energy production. The process involves the carnitine fatty acyl carrier system. First, on the outside of the inner mitochondrial membrane, we have an enzyme called carnitine acyl transferase 1. This enzyme catalyzes the reaction between acetyl-CoA and carnitine, forming acyl-carnitine and releasing CoASH. Next, the acyl-carnitine is transported across the inner mitochondrial membrane by a transporter called carnitine acyl translocase. Interestingly, as acyl-carnitine moves into the mitochondrial matrix, free carnitine is transported back out into the cytosol, maintaining a balance. Finally, once inside the matrix, another enzyme called carnitine acyl transferase II reconverts acyl-carnitine back into fatty acetyl-CoA, ready for beta-oxidation, and releases free carnitine. This entire process effectively shuttles fatty acyl groups into the mitochondria, which is a crucial step in harnessing fatty acids for energy. So, the carnitine fatty acyl carrier system relies heavily on the presence of CoA on both sides of the inner mitochondrial membrane. Essentially, for fatty acids to be broken down efficiently, they need to be transported into the mitochondria in a specific way. This process depends on CoA being available both inside and outside the mitochondria. When fatty acetyl-CoA molecules are ready to enter, they first need to be converted into a form that can cross the mitochondrial membrane. This is where the carnitine shuttle comes into play, helping transfer fatty acids into the mitochondria so they can be oxidized for energy. It's a finely tuned system that highlights how vital CoA is for energy metabolism, especially in the context of fatty acid breakdown. In the mitochondrial matrix, fatty acetyl-CoAs undergo a process called beta-oxidation, where they are broken down into acetyl-CoA molecules. This happens through a repeating sequence of 4 reactions, each one removing a 2-carbon unit from the fatty acid chain. Think of it as a cutting process that systematically shortens the chain, releasing energy along the way. The key point here is that this process is highly efficient and is the primary way our bodies extract energy from fats. As we follow these reactions, the acetyl-CoA produced can then enter the citric acid cycle to generate even more energy. Connecting this to the overall theme, beta-oxidation is fundamental for understanding how fats serve as a major energy source, especially during fasting or prolonged exercise. It's a beautifully coordinated process that highlights the body's ability to harness stored fat for power. Now, let's talk about oxidation. When fatty acetyl-CoA enters this stage, it's oxidized by a specific enzyme called acetyl-CoA dehydrogenase. This enzyme plays a crucial role because it initiates the process of breaking down fatty acids to generate energy. Essentially, it helps convert the fatty acetyl-CoA into a form that can be further processed in the mitochondria. Next, let's consider how fatty acetyl-CoAs get into the mitochondria in the first place. This is a key step because without proper entry, the entire process of fatty acid oxidation can't proceed efficiently. The mitochondria have specialized transport mechanisms to facilitate this entry, ensuring that fatty acids are available for oxidation when the body needs energy. Understanding both the oxidation process itself and how fatty acids are transported into the mitochondria connects to the bigger picture of how our body efficiently harnesses stored fat for energy, especially during periods of fasting or increased activity. Let's take a closer look at the mitochondrion and its role in fatty acid oxidation. Inside the mitochondrion, there are at least 4 specific dehydrogenases that target fatty acetyl-CoAs of different chain lengths. These are known as very long chain, medium chain, and short chain acetyl-CoA dehydrogenases, abbreviated as VLCAD, LCAD, MCAD, and SCAD. VLCAD is responsible for oxidizing straight-chain acetyl-CoA molecules ranging from C12 to C24. Meanwhile, MCAD has a broader range, but is most active with substrates around C6 and C8. As for SCAD, it prefers C4, then C6, and C8. LCAD, on the other hand, plays a key role in initiating the oxidation of branched-chain fatty acids. Understanding these specificities helps us see how the mitochondria efficiently process fatty acids of various chain lengths, which is essential for energy production. Now, let's also briefly touch on how fatty acetyl-CoAs enter the mitochondria for this process. Now, let's talk about the hydration step in fatty acid oxidation. When the unsaturated fatty acetyl-CoA is processed, it's hydrated by an enzyme called enoyl-CoA hydratase. This enzyme adds a water molecule, converting the double bond into a beta-hydroxyacyl derivative. It's important to note that these hydratases are quite specific. They show chain length specificity, meaning they work most efficiently with certain fatty acid chain lengths. This specificity helps ensure the process proceeds smoothly and efficiently. Overall, this hydration step is crucial because it prepares the molecule for the next stage of oxidation, linking the initial entry of fatty acids into mitochondria with the subsequent breakdown steps. Now, let's look at the next steps in the beta-oxidation process. First, the oxidation step involves the conversion of the beta-hydroxy derivative into a beta-ketosylCoA. This is carried out by the enzyme beta-hydroxyacyl-CoA dehydrogenase, and during this reaction, NAD+ is reduced to NADH. This step is crucial because it prepares the molecule for the subsequent cleavage. Following that, we have the thiolysis step. Here, a CoA molecule comes in and cleaves the bond in the beta-keto derivative. This reaction results in the formation of acetyl-CoA, which can enter the citric acid cycle, and a shortened fatty acyl-CoA, ready to undergo another round of oxidation. This cleavage is catalyzed by the enzyme thiolase. These steps, oxidation and thiolysis, are vital because they systematically break down fatty acids into acetyl-CoA units, fueling our energy production. It's a finely tuned process that highlights how fatty acids are efficiently transformed into usable energy within the mitochondria. Now that the shortened fatty acid chain is prepared, it's ready to go through the next cycle of beta-oxidation. A key point here is that when dealing with long-chain fatty acids, the oxidation process involves a special complex. This complex is made up of 3 enzymes: acetyl-CoA hydratase, 3-hydroxyacetyl-CoA dehydrogenase, and beta-catathiolase. What's interesting is that these enzymes don't act independently. Instead, they form a membrane-bound trifunctional protein. This setup is unique to long-chain fatty acid oxidation. In contrast, when we look at medium and short-chain fatty acids, the enzymes responsible for oxidation are different. They are soluble proteins located in the mitochondrial matrix. So, the overall picture here highlights how the body has specialized machinery depending on the chain length of the fatty acids we're working with, emphasizing the efficiency and specificity of mitochondrial fatty acid metabolism. This distinction is important because it shows how the mitochondria adapt their enzymatic complexes to handle different types of fatty acids, ensuring smooth and efficient energy production. Remember, understanding these specialized complexes helps us appreciate the intricate regulation of fatty acid oxidation within the mitochondria, which is central to overall energy metabolism. Now, let's look at what happens during each cycle of beta-oxidation. Each cycle produces 1 FADH2, 1 NADH, and 1 acetyl-CoA. This is important because these molecules are key carriers of energy. During the final cycle, instead of producing 1 acetyl-CoA, 2 acetyl-CoAs are generated, which makes sense because we're breaking down a larger fatty acid into smaller pieces. Putting it all together, for the complete oxidation of palmitate, we end up with 8 acetyl-CoA, 7 FADH2, and 7 NADH molecules. These are the main energy carriers generated from one molecule of palmitate. Now, each FADH2 and NADH can be oxidized in the electron transport chain. FADH2 yields about 1.5 ATP, and NADH yields about 2.5 ATP. Meanwhile, each acetyl-CoA, when it enters the TCA cycle and undergoes oxidative phosphorylation, produces approximately 10 ATP. When we total all of these, the complete oxidation of palmitate results in roughly 108 ATP molecules. But there's a catch. Activating the fatty acid to start this process consumes 2 high-energy phosphate bonds, which is equivalent to 2 ATPs. So, to get the net yield, we subtract those 2 ATPs. Therefore, the net ATP yield from the oxidation of palmitate is about 106 ATP molecules. This highlights just how efficient fatty acids are as an energy source, fitting perfectly into our overall understanding of cellular metabolism and energy production. Let's take a closer look at the beta-oxidation pathway. This process is essential for breaking down fatty acids into smaller units that our bodies can use for energy. Essentially, beta-oxidation occurs in the mitochondria, where fatty acids are systematically cleaved. The key idea here is that each cycle shortens the fatty acid chain by 2 carbons, producing acetyl-CoA, which then enters the citric acid cycle for further energy production. It's important to note that beta-oxidation is a highly regulated pathway, especially during fasting or prolonged exercise, when the body shifts to burning fat instead of carbohydrates. This pathway is a cornerstone of our overall energy metabolism, linking dietary fats to usable energy. Understanding it helps us see how the body efficiently adapts to different energy demands, which ties back to the broader theme of metabolic flexibility. Now, let's take a closer look at the energy difference between the oxidation of palmitoyl-CoA and palmitoyl-CoA. As you can see here, the key point is that the degree of saturation in the fatty acid chain significantly influences the amount of energy we can extract during beta-oxidation. Palmitoyl-CoA, which is a saturated fatty acid, tends to produce more energy compared to palmitoyl-CoA, which contains a double bond. The presence of that double bond in palmitoyl-CoA means that one of the steps in its oxidation pathway is slightly different or less efficient, leading to a small reduction in total energy yield. This difference underscores an important concept in lipid metabolism. Unsaturated fatty acids, due to their double bonds, generally yield slightly less energy per molecule than their saturated counterparts. Overall, understanding these nuances helps us appreciate how the structure of fatty acids influences their metabolism and energy production, which is central to our broader discussion of metabolic flexibility and energy management in the body. In this session, we have explored how the utilization of fatty acids for energy production varies quite a bit from one tissue to another. It really depends on the specific metabolic needs of the tissue or organ at any given time. We also looked at how and when this energy is used, which helps us understand the dynamic nature of metabolism in different physiological states. Throughout this study session, you've learned about several key concepts. First, the process of fatty acid activation, which is the initial step that prepares fatty acids for breakdown. Then, we examined beta-oxidation of fatty acids, the pathway through which fatty acids are catabolized to generate energy. Lastly, we discussed the net ATP yield from palmitate oxidation, giving you a clear picture of how much energy can be derived from fatty acids. In conclusion, understanding these processes highlights how crucial fatty acids are as an energy source, especially in tissues like the heart and skeletal muscle, and how their utilization adapts based on the tissue's metabolic state. This knowledge ties back to our overall theme of metabolic flexibility and energy management in the body. Let's wrap up by discussing why fatty acids are more efficient than carbohydrates as fuel molecules. Fatty acids, like palmitate, provide a much higher yield of ATP per molecule because they are more reduced and contain more carbon atoms, which means more energy can be extracted during oxidation. This makes them an ideal energy source for long-term, sustained activities. Now, regarding palmitate oxidation, the net ATP produced is approximately 106 molecules. This includes ATP generated during beta-oxidation, the citric acid cycle, and oxidative phosphorylation, minus the costs of activation and transport. Speaking of activation, fatty acids are activated in the cytoplasm by attaching to CoA. Forming fatty acetyl-CoA, this process requires ATP and is catalyzed by acetyl-CoA synthetase. This activation step is crucial because it prepares the fatty acid for subsequent transport into the mitochondria and eventual oxidation. Connecting this to our overall theme, understanding these energy pathways highlights how the body efficiently utilizes different fuel sources, especially during fasting or prolonged exercise, emphasizing the importance of fatty acids in energy metabolism.